This protocol will help researchers more accurately determine the native oligomeric state of membrane proteins by utilizing the native cell membrane nanoparticle system in conjunction with electron microscopy. The advantage of this technique is that it provides accurate structural data on membrane proteins in a native cell membrane-like environment and can be utilized for future high-resolution structure determination. Demonstrating the procedure will be Kyle Kroeck, a postdoctoral fellow from my laboratory.
To prepare native cell membrane nanoparticles, resuspend 1 gram of the membrane pellet of interest in 10 milliliters of NCMN buffer A. Use a glass Dounce homogenizer to homogenize the resuspended cell membrane sample at 20 degrees Celsius and transfer the suspended sample to a 50 milliliter polypropylene tube. Add membrane-active polymer stock solution and additional NCMN buffer A to the sample to a final concentration of 2.5% membrane-active polymer, and shake the solution for 2 hours at 20 degrees Celsius. At the end of the incubation, ultracentrifuge the sample. During the centrifugation, equilibrate a 5 milliliter nickel NTA column with 25 milliliters of NCMN buffer A. At the end of the centrifugation, transfer the supernatant onto the column and use a syringe pump set to a 0.